So in this module, we want to talk about the basic electrophysiology of the brain. So in particular, we want to talk about some very basic things. So electric charge and fields, because that's what we're really dealing with, is charge, electric charge. We want to talk about voltage as a measure of energy, because we talk about voltage, but what does it mean? And when we talk about what we measure, we're measuring something in microvolts, but we're measuring something to do with charge rather than some abstract idea. And ions as charged particles, because these are really what we're dealing with. The body's a big sack of charged particles floating in water, basically, and that's what the brain is also. Then we're going to talk about properties of neurons and neuronal membranes, membrane potentials, action potentials, synaptic potentials, and volume conductive potentials to the scalp. So this kind of lays the foundation for all the things that we measure. And if you understand these concepts, which we've all studied in medical school or somewhere, then what happens with the data is clearer than if you just think of the data as the epiphenomenon. So that's the objective of going through this material. There's a number of basic ideas to try to understand. One of this is this concept of electric charge, and the fundamental definition of what a charge is a charged particle and another charged particle. You know they're charged because they produce forces on each other. There's not a direct concept of what charge is. Charge is something which produces a force on something else which has charge. So that's an important thing to have as an idea. And as you probably all remember, electric force is charge experience as a force produced by another electric charge. And that was Coulomb's law, which probably most people studied in physics or in their basic physiology courses. So Coulomb's law is this F equals some constant. We don't need to worry about what the constant is, and then the product of two charged particles, okay, divided by the square of the distance between them. This is kind of like one of the fundamental laws of physics, and that's measured in newtons. So force is measured in newtons. The charged particle is an electron or an ion, and the charge on one electron is 1.602 times 10 to the minus 19th coulombs. So Coulomb is a name we've assigned to charge. And charge is defined by this force equation. So, sodium, potassium, calcium, they're things we think about every day, and they're charged particles. They happen to have a positive charge, which is the absence of electrons. And then chloride is a negative charge. It has a charge because it has a surplus of an electron. So an atom normally is neutral. An atom of a substance is neutral, but the absence or the excess of an electron is what gives its charge. So these are important ideas to have in your mind, and it seems like it's distant from neurophysiology, but it's really the basis of all of neurophysiology. And of course, electrons are governed by the law of conservation. So it's one of the four laws of conservation in physics, is that the number of electrons are conserved within a system. So really important ideas. Uh, I just want to reinforce the idea of an electric field. So if you think of two charges and they have a force between them, okay, then the way the physicists have tried to describe this is with the idea of an electric field intensity, which is the force per unit test charge. So if we go back to this, I don't go back that far. Force was the product of two charges divided by something. Then electric field intensity is that force divided by one of the charges. So you can think of, you got a charged particle sitting here, and it's got this field radiating out from it, and that's called an electric field. So conceptually, that's what the map would look like. So you've got a positive charge, and they represent the field as going out from it, and a negative charge field is coming in. Really basic concepts, but they drive all of physiology. And the units, you can tell I'm really big on units by the time we get done. The units for electric field intensity is newtons per meter. So it's the force distributed across the distance. So potential energy, which is what we're really interested in, which is the work. So potential energy is work. So moving something from here to there, you do work, and that we describe either as potential energy or kinetic energy. So potential energy is the work done in moving a unit charge from one point to another in an electric field. So imagine we've got this charge here, and we've got a field radiating out from it, and you take another charge and you move it in against the electric intensity produced by this charge. That's doing work, and so you're capturing that work in those two particles. That particle coming in is capturing the work. So the basic definition of work is force times distance. So that's Newton meters. That's the units of work. Force times distance is newtons is force, meters is distance, and we've given that a name called joules. So joules is just a name for work. And then the electric potential, what we call volts, is just joules divided by charge. So the concept behind all these things is that it's energy related to charge, joules per coulomb, and we call that a volt. So when we're talking about a volt, what we're talking about is the capacity, ability within the nervous system for particles to move and do work. And that's what they're doing. When the particles move, they do work. And so this then becomes the basis for everything that happens. That's why you need oxygen. We'll talk about that. Where's the oxygen consumed and all that within the nervous system. So these are basic ideas that help you understand when you see changes, what's happening. It seems like they're distant from the epiphenomenon of an EEG or an evoked potential, but that's in fact what you're looking at. So electric current. So an electric current is defined as a moving electric charge. So we have these particles that can be an electron or they can be an ion, but when they move, we call that a current. And That's the time rate of change at which a charge crosses a surface. So conceptually, if you think of it as you've got some surface and you've got a charge flowing across that because there's some force field on the charge, and that's producing a current. And again, current I is the time rate dq dt, the time rate of charge across a surface. Charge, remember, was coulombs, so that's coulombs per second, and that's an ampere. So that's and those are the units you use all the time. You talk about amperes and volts, and that's what they mean. They don't mean, volts has no meaning aside from these concepts. So that's what we're getting at. So 1 amp is 1 coulomb per second, and it's 6.24 times 10 to the 18th electrons per second, or charges per second. That's a big number, very, really big number. 1 milliamp, we talk in physiology, we talk more about milliamps and millivolts. Still a big number. It's 6.24 times 10 to the 15th electrons or charged particles per second. So, when we're talking about, we think there's a milliamp of current, we're talking about an awful lot of charged particles moving across some surface. And it's kind of important to keep these ideas in your mind. At the end of the day, you can look at the data and you can interpret it, but if you understand these things, you'll understand better about what the data is telling you about what's going on in the underlying physiology. So just to reinforce some basic ideas, electrons flow in a metal. So you need a metal, you need a conductive metal of some kind to have electrons flow. Charged particles, charged atoms, which we call ions, they move in salt water. So that's what we have in our body. We have charged ions moving. Now they're a little different than electrons. The charge concept is the same, but the ions have a larger mass, thousands of times larger mass. So some of the terminology you come up with is an anode. An anode is a plus terminal, and it's a sink for anions. So anions, which are negatively charged particles, will flow into an anion, and it's a source for cations. So positive charged particles will flow out of an anode. Yeah. Cathode works the opposite. It's a sink for cations and a source for anions. So those terms will come up all the time, and so it's kind of important to have it clear in your mind what we're talking about. Now, there's two other important concepts. One is what's called an electric dipole. An electric dipole is, again, if you go back and you envision two charges, and they're separated by a distance. People call that an electric dipole. So, and that has the dimensions of coulomb meters. And there's another idea called a current dipole, which is two current sources separated by a distance, and there's current flowing between those two sources. Of course, current always has to flow in a loop. So there always will be some return path for that current. But the concentrated current flowing between two current sources, people call a current dipole. And this drives all of electrophysiology. And the units for that are an ampere meter, an amp meter. So it's charges per second flowing through a distance. Very physical kind of stuff. This is a good place to stop for a second and see, are there Any questions about that? It's kind of basic physics, but it creates, physics and physiology aren't that far apart. So, anybody have any questions? Okay, so we're going to kind of change direction slightly. We're going to take those ideas and apply them to neurons, which is, of course, what we're interested in. How do they apply to neurons? I think we all remember the fundamental idea of a neuron, which is a cell, stereotypic cell that has dendrites, that has a cell body, it has an axon, it has terminal boutons, and these neurons are interconnected through the synaptic junctions. So that's the fundamental thing, and that's what we're going to apply those ideas to families of neurons, to look at how EEG and evoked potentials are generated, and then it makes sense. That drives a way you can think about the changes that are occurring in these epiphenomena we call EEG and evoked potentials. So everybody, I'm sure, remembers the cerebral cortex. It's a basically 3 millimeter thick layer of neurons. It's divided into 6 layers, layer 1 through layer 6, and the key neurons in the cerebral cortex are the pyramidal neurons. The pyramidal neurons have inputs that flow into them, synaptic inputs that flow in. They come from the thalamus, and they come from other areas of the cortex. So they're part of a large interconnected network. And people understand that different of these inputs come in at different levels. So some inputs will come in at level 1, some inputs come in at level 4 in the cortical mantle. But it's important to have that clear in your mind, that you've got these pyramidal neurons which are orientated perpendicular to the cortex, and that's their stereotypic orientation. And it's important to understand that, because that's how the physics we just talked about will map onto the electron, onto the neurons, and then will produce voltages or electric fields, if we're measuring fields, that we talk about. So the properties of the neuronal membranes are extremely important to have some underlying idea of how they work. And everybody has seen this in their physiology courses, but I wanted to re-emphasize this over again. You have a membrane which is selectively permeable to ions. So on one side you have a preponderance of potassium on the inside and anions, proteins. And on the outside you have a preponderance of sodium. And it's the fact that the membrane is selectively permeable to these ion concentrations which produces the voltages which drive everything that we're interested in. There are a number of different kinds of proteins. We're not going to dive into that deeply. It's just important that that you think about them, and that the properties of the synaptic junctions that we're going to deal with are dependent on the properties of these proteins, because we're interested in the class of synaptic junctions called inotropic synapses, namely they use ions to generate the potentials that we're interested in, and their properties are governed by the fields that are set up across the membrane and by the changes induced in those fields by other chemical transmissions that I'm talking about. So these are important ideas because they drive the generation of the EEG and blood potentials. So this kind of recapitulates most of what I just said. There's ion concentrations. The outside is heavily sodium dependent. There's chloride. Inside is potassium and anions that are protein based, organic ions. This sets up the potential differences, which are captured in the equation form in the Nernst equation, which we'll talk about in a second. There's an extracellular excess of positive charges, and there are forces. So forces that we talked about, that's what drives everything with the forces. The membranes allow diffusion to areas of low concentration, so sodium will continually leak through the membrane. Potassium will continually leak out of the membrane. That's measured with a parameter called conductivity, or more fundamentally, mobility. We don't have to worry about what those numbers are. We just have to realize that that's going on, and that there's this sodium-potassium pump which works to correct that leakage. So as you have sodium leaking in and potassium leaking out, then there's the sodium-potassium pump, which is continually trying to correct that. The understanding that the sodium-potassium pump is there is very important, because the brain takes a very large percentage of the total energy consumed in the body. And 2/3 of the energy consumed in the brain is consumed by the sodium-potassium pump. So when you see a change in a physiological measure, most of the time what you're seeing is a change in the sodium-potassium pump being starved for energy to drive the pump. And we'll talk a little bit about what the bioenergetics are, just so you appreciate the importance of that, because it speaks to, as we'll talk about later on, it speaks to the importance of oxygenation and blood pressure, cerebral perfusion pressure, these ideas which end up providing the blood, which carries the nutrients, which drive the pumps, which produces the signals that we're looking at. So you kind of got to have this roadmap in your mind. You don't have to know the numbers, but you gotta conceptually have this, so when you see changes that are related to changes in blood pressure or perfusion, you have an idea of what's happening, and you know why, as Dr. Clyde mentioned, if puts a temporary clip on and he has a change, he knows he's got 3 to 5 minutes or 6 minutes where he can work. If you don't see a change, you know you can work for forever, basically. So it's because these things aren't slammed. They're not necessarily, they don't go to zero immediately. They have energy reserves, but the energy reserves are very small because the brain uses so much energy. So this is very important to have this kind of concept in your mind when you think about the signals that you measure. Okay, this is just again to re-emphasize some important ideas. The this cell membrane, which is conductive for potassium and sodium and chloride. So you have a neuron that has an internal membrane potential, which typically sits somewhere around minus 70 millivolts. So that's the potential inside the neuron. Potential is always measured between 2 points. So if I tell you I've got a voltage of something, it's a voltage of something measured against the voltage somewhere else. So there's always got to be 2 points. So when you measure the potential across the membrane, you're measuring inside the membrane against outside the membrane. And typically that's minus 70 millivolts. That is controlled by potassium. So if you look up here in this table, so if you look up in this table, here's potassium. It tells you what the concentration is, extracellularly, intracellularly. And then what Nernst equation would tell you that resting potential is. And it's about at minus 70 millivolts. You remember what a volt is? It's, joules per coulomb, Newton meters per coulomb. So it's the amount of energy you have trying to push something in a certain direction. Sodium, on the other hand, has a resting potential. It should be about plus 60 or plus 80 millivolts. So measuring inside to outside, the resting potential for sodium would be plus 80 millivolts. So it's got this energy trying to force you in the other direction. Typically, these potentials are separated by the membrane. Now, we've got chloride on here, which at this point we don't care about. Chloride's important, and we'll talk about it later on, but for the purposes of this discussion, it's not as important. And there should be calciums on there, and again, calcium isn't that important for purposes right now. So within, again, here's a different view. Here's the extracellular, here's the cytoplasm, the intracellular. Potassium is the predominant ion, and we have these anions, which are primarily proteins. And you have calcium leaking out, you have sodium leaking in, and you have the sodium-potassium pump, which is pumping out 3 sodium ions for every 2 potassium ions that come in, and it's using up ATP in the process. And that's where I want to reiterate that because it's important to understand. That's where 2/3 of the energy used in the brain is used, is in maintaining that sodium-potassium pump. And almost everything we talk about in neurophysiology is related to usage of energy. It's related to trying to maintain those ion concentrations in the equilibrium state, or trying to recover the equilibrium state after something happens. So, properties of action potentials. Of course, we think of action potentials as, at one level, we think of them as an all or none event. Something happens, the neuron fires, some very brief event occurs, and it goes on. Well, there's a number of things that are important. One, the base of the action potential, so this line here, that's controlled by the potassium equilibrium potential. So remember, potassium is the predominant cation inside the cell. So the action potential can only go as low as the potassium potential. So that may be, in this example, it's minus 85 millivolts. It could be something different. The peak of an action potential is controlled by the sodium equilibrium potential. So sodium is the predominant ion outside the cell. And so those are the boundaries between which an action potential can occur. Minimum value defined by the potassium value computed from the Nernst equation. Peak value from the sodium value computed from the Nernst equation. And then you get the shape of the action potential, which is related to, if you look at these two curves here, this is called sodium conduction. This one's potassium conduction. So when you reach threshold, which we would call this minus 70 millivolts, which is really a function of the chloride and the ion and the protein ions and the potassium ions, but you reach that threshold, the sodium conduction starts to open up. And the idea behind the conduction is it's like a resistance. It's a conductance. So conductance is the inverse of resistance. So the property of the membrane changes as a function of the voltage across it, and the ions flow through it. So the inflow of the sodium ions drives the transmembrane potential in a positive direction. It can't get all the way up there because it's really not pure sodium, it's a mix of things. Okay. And then the potassium channels start to open, and they start, as this sodium conductance peaks, the potassium conductance is kicking up, and that pulls you back down towards the potassium equilibrium. So this is what's going on with the action potentials. Now, we normally don't measure things which are directly related to action potentials. What we measure are things that are related to excitatory postsynaptic potentials or inhibitory postsynaptic potentials, which are driven by action potentials. So it's kind of important to keep that in mind, that everything we measure, not everything, but most of the things we measure are driven by the synaptic potentials. And hopefully we'll put this all together so it makes sense. So the synapses are the key to everything. And we're particularly interested in these what are called ionotropic synapses, namely where gates are open, ions flow, and they're fairly quickly active. There's another group of synapses called metabotropic, which work over many, many seconds or minutes. So these are fast, in the order of milliseconds. They change the postsynaptic membrane polarity. So remember, we're measuring the potential across the membrane. These potentials will then change those potentials. So they produce a postsynaptic membrane polarity change. They'll depolarize it, even with dry towards firing, or they'll hyperpolarize it. They'll pull it away from firing. And they produce focused synaptic connections. And there's a whole bunch of neurotransmitters that drive this phenomenon. Acetylcholine, GABA, glutamate, glycine. These are the things which the anesthesiology folks play with, is the effect of their drugs on these transmitters, which then change the properties of the neurons that we're trying to record from. So it's all a big integrated mishmash, basically. So, any questions so far?
Мы хотели бы пообсуждать калий-натриевый насос, фундаментальные основы формирования всех видов потенциала, о которых сейчас нам рассказали.
It's a dangerous topic for us.
Yeah.
It's much comfortable for us just listening.
It's a topic which you don't want to spend. You can spend a whole day on it, and you don't want to do that because you want to get on to other things. But you've got to have an idea because it changes how you think about everything.
It's very important to keep in mind principles.
Yeah. So here's our brain, and we want to talk now about EEG generators. We're going to kind of go over the biophysics of that, and in a later section we'll actually talk about it from the EEG perspective. But I want to tie this together at the biophysics level, at the biophysics level, because it makes a lot of things clear if I can get the concepts clear. So we talked about the cortical layer, and this is the cortical mantle here represented in this diagram. And these are sulci, and then there's CSF, and there's skull, skin, and then eventually you get to an electrode. Here are pyramidal cells, and they're, as I said, oriented radially towards the surface of the cortex. So that's the fundamental property of a pyramidal neuron. They're always radial with respect to the surface of the cortex. So you can see these neurons are pointing in that direction. These neurons are pointing in that direction. These neurons are pointing in this direction, and so forth. That's extremely important to keep that idea in your mind, that whether you work in EEG or you work in the operating room or you work in the diagnostic lab, it's important to keep that in your mind. And no matter where you are on the cortex, it's pretty much any neocortical area has that standard feature to it. So older areas of the cortex don't, or if you're in the hippocampus, it has a different set of properties. But in the neocortex, this is what we're dealing with. So here's our 6 layers of the cortical mantle again. And remember, this cortical mantle is only 3 millimeters thick. That's not thick. You know, we look at these pictures and we think, boy, it's really thick, but it's a very thin mantle. And so all the activity we're recording, most of the activity we're recording is being generated within that cortical mantle. And again, the pyramidal neurons are organized radially, so they project the cell bodies are about in this region, and then this shows you where the inputs come from. From the thalamus, it's very important. The thalamus drives the rhythms of the cortex. And so this input information is flowing in continuously. So what happens in the neurons? So these are supposed to represent the pyramidal neurons, and they show some input flowing in at level 1 or 2, some input flowing in at level 4. And they produce different kinds of effects. But they produce in the same way. Remember, the transmission from one neuron to the next neuron is through a synaptic junction. And the synaptic junction will produce an excitatory postsynaptic potential or an inhibitory postsynaptic potential. Could produce either one. So look at this bottom figure. This is coming into layer 4, and it's excitatory input. Now, if you look at this figure, these are minus signs on the output of the neuron. So remember, when we talk about the transmembrane potential, we're referencing the inside of the neuron to the outside of the neuron. So we're measuring the potential across the membrane of the neuron. Okay? So when you produce an excitatory postsynaptic potential, an action potential flows in, hits this synaptic junction, it's going to produce an EPSP. The EPSP is a depolarization of the membrane potential inside the neuron. So that becomes more positive inside the neuron. So the neuron's normally at minus 70 millivolts, it may go up to minus 65 millivolts. It becomes more positive, the effect of the EPSP. But that's happened because positive charge has flown into the neuron. But when positive charge flows in, negative charge accumulates outside the neuron. So these minus signs represent negative charge accumulating outside the neuron due to positive charge moving into the neuron. So there's some steps here. Then what happens is, since this is negative charge accumulating here, positive charge will accumulate at the apical region. So we call this end of the neuron the apical region. So you've got negative charge here due to the positive charge inside, which produces positive charge here, and that's recorded at the scalp as a positive voltage. And this particular figure, positive is plotted downward. So this is a very complicated set of ideas, but it drives everything. And if this is clear, if we can make this clear, then you'll understand EEGs, evoked potentials, and everything, because it all comes out of this. So remember conservation of charge, we talked about that. So the physics of the system is always going to try to maintain a balance in charge, and it will balance it. So you get an EPSP here, the inside in here becomes positive, the outside therefore becomes negative. That produces positives up here, which are seen at the scalp as a positive voltage. Now, the other key idea, remember, we talked about was if you have distributions of charge, you will have current flow between them. So there's a current that will flow from here to here, and that's actually what we're sensing on the scalp, is that current which is flowing. We call that volume conduction. And then there's a current that flows down the center of the axon, and that's that current dipole we talked about. Okay, so current flows through the center, actually the center of the dendritic structure, to try to equalize the charge, and it returns out here, because current always flows in a loop. Voltage is always measured between 2 points. Current always flows in a loop. So you've got these charges accumulating due to the EPSPs or the IPSPs, and you can see it doesn't matter. The projections come in here from the thalamus, from here from the thalamus, here from the other parts of the cortex. They're going to set up either EPSPs or IPSPs, and what we see then is the effect of these things. Very, very important to try to understand this. So, just to reiterate it, because I think it's so important. Pyramidal neurons oriented radially with respect to the cortex. You have synaptic inputs coming in. Let's just say at 2 layers, layer 1 and layer 4. They will set up either EPSPs or IPSPs, and therefore these will set up currents, which flow is represented in this diagram. They'll flow diffusely through the extracellular fluid and in a very concentrated form through the intracellular fluid. So this is what produces the things we measure. This is what produces the EEG and the evoked potentials we measure. There's nothing mysterious about it. It doesn't appear out of nowhere. It's this physical phenomenon which is what's producing it. So I want to drive this idea of volume conduction home. Everybody in this room knows what volume conduction is. Everybody has done an EKG. An EKG, which you measure on the surface of the body, is a volume conducted signal from the heart. What that means is the heart pumps, produces an electrical gradient which causes the ions to flow and which spread out across the surface of your body and then come back somewhere else on the surface of the heart. And that's volume conduction. It's exactly the same idea applied to these pyramidal neurons. So the neural activity is conducted by volume conduction to the scalp and the sensors. Volume conduction is linear. And what that means is, if you've got a neuron here and a neuron here, and they overlap and they're both producing volume conduction, what you see will be exactly the sum of those two signals. So it's either linear in the world or it's nonlinear. If it's nonlinear, it's not the sum, and if it's linear, it's the sum. That's the basic definition of that. And each measures a weighted sum of many neurons' activities. So in this schematic, what I've shown is you got to measure voltages between 2 points. So you got a potential here, you have a potential here, and you're seeing the difference between those two, measuring one against the other. Each of these represents a weighted sum, I'll tell you what I mean by that in a second, of a lot of neurons synchronously being activated. Okay. A weighted sum means that I've got a neuron here producing something, and I've got a neuron here producing something, and my electrodes here, even though what the electrode will see will be a linear sum of the two, the strength of this guy will be more than the strength of this guy because this neuron is closer to the electrode. So that's a weighting factor. So depending where you are in the cortical mantle, remember there's 6 layers. Several of the layers have these inputs that excite them or inhibit them. You're going to get a difference in the weighted sum of this activity. So it's an important thing to try and understand in terms of how this fits together. The other thing I wanted to mention at this point, because you see these words come up all the time, near field versus far field. Remember, we talked about what a field is. But there's some important points that underlie this. One, biological tissue is an electrically conductive material. Remember, I told you basically the body and the brain is a sack of water full of ions separated by membranes. That's basically what everything is. So we're a big sack of water with tons of ions in it, and the water is conductive. These ions they're dissolved in the water, and they will migrate in the directions in which the forces push them to migrate. And the forces come from the electric field, and they can come from the permeability of the membranes also separating these things. If all tissues had the same resistance current would flow equally in all directions. So if we were truly just a bag of water, then everything would flow homogeneously. But we're really not quite one bag of water, we're a bunch of bags of water, so things flow not homogeneously. And that's where the idea of resistance comes in. So the idea of a near field response and a far field response is based on some simple ideas. One is the location of the generator. The second one is the location of the recording site. So people will say, oh, that's a near field signal. That means that the generator site is close to the recording site. That's all it means. The amplitude and the orientation of the potential may be important because remember, these pyramidal neurons are orientated radially to the cortex. They're always radial to the cortex, but the cortex is very complicated in its geometry and its topology. It rotates in many, many directions. So it's important to realize that one segment of cortex can be projecting a signal in this direction, and a segment not too far away from it can be projecting a signal in this direction. And you may not see what one part of the cortex is doing when you can see another part. So even though people use this terminology, excuse me, even though people use this terminology and they use it very rigidly, they'll say, that's a near field potential, that's a far field potential. There is no acceptable definition. It's more of a concept. Something is close to the generator or something, or the recording electrode is far away from the generator. And that's the important thing. So the way I try to make the point is a far field potential is an EKG because you're far away from the generator by definition. A near field potential would be the EEG because, or say the cortigram because you're sitting right on the cortex, so that would be a near field. And that's pretty concrete. You can't make it much more separately than that. So there's some important numbers here. We talked about, we conceptualized pyramidal neurons as single neurons, but it's in fact, that's not what's happening. And we talked about synaptic junction and a synaptic junction to make it very specific, but the estimates for pyramidal neurons are that there are on the order of 100,000 synaptic junctions on each pyramidal neuron, 100,000. So that's a big number. The dimensionality is high. It's been estimated that it takes approximately 50,000 pyramidal neurons being activated synchronously to produce a detectable signal on the scalp. So if you think about the dimensionality, the numbers of what we're talking about, you've got to have on the order of, doesn't mean exactly, but on the order of 50,000 of these radially orientated neurons activated synchronously through the inputs of coming in through 100,000 synaptic junctions to produce a signal that we're going to record and call the EEG or the evoked potential. So that's the physics of this thing. This involves approximately, let me go back for a second. For the EEG, that's approximately a square centimeter of brain tissue. Now, there's the idea of columnar organization of the cortex. So the cortex tends to be organized in columns. It turns out the columns contain several thousand neurons, 10 to 20,000 neurons. So the numbers kind of agree that people have come up with from different calculations and different experiments. So this is what you're dealing with when you think about what I'm trying to measure. Now, I made a comment on here and I said with today's technology, because as technology gets better, and we'll talk a little bit about that, as technology gets better, then you can reduce the number of neurons that have to be synchronously activated to produce a detectable signal. And the fewer neurons you have to have activated to produce a detectable signal, the better you can be about saying how are these things being affected by the operation. So this fits together in terms of trying to understand the data. Let me kind of summarize the stuff this way. Synaptic input. Here's our cortical mantle. Synaptic input is excitatory or inhibitory. There are synapses, but the dendrites are close to the cell body, so at layer 1 or 2, or say at layer 4. These will set up these current flows. In this case, it's the current dipole being represented. The current flows down through the dendritic structure and then diffusely back through the extracellular volume. People have estimated that it takes about 1% of the neurons being synchronously activated, which is about 50,000 neurons to produce a detectable signal. That work was done by Rita Harry, who's actually in Helsinki. I think she's still working. She's a physicist. And it's dependent on the orientation of the primary current. So, if you have we think about conductors as being perfectly shaped. In our case, we're dealing with the cortical mantle, the brain, which isn't perfectly shaped. It's got all these convolutions, these sulci, and so things are, even though the neurons are radially oriented towards the surface, they could be pointed almost, in a lot of directions because the sulci are very curved and the cortical mantle is curved. So what we're seeing is some kind of summation of the radial tangential of all these currents being generated. Again, this kind of summarizes it. We've got a pyramidal neuron here. The light lines are the electric field intensities producing the voltages, which you record between 2 points on the scalp. So each area of the scalp is being excited in a different way than another area on the scalp. You're recording those potential differences. I know Brett's kind of interested in the MEG. MEG is produced by exactly the same phenomenon, but it's produced by the currents flowing through the pyramidal neurons directly, and that produces these extracranial magnetic fields. So different views of the same neurons doing the same thing. So summary, there's a lot of very interesting stuff here, but it's not something that'll make sense to you immediately unless you read it over. So the basic ideas of what charge is, the definition of charge, I mean, it's the way it's defined by the physicists is the force produced by charged particles. That leads us to the idea of force fields, electric intensity. Charge can be on electrons, which is the basic phenomenon we call electrons, or it can be in an ion, which is a charged particle, which is either the positively is the absence of an electron, negatively it's the excess of an electron. So we have anions and cations. These then play into the cortical layer. We have the neuronal mechanisms of ion concentrations. resting potentials and sodium potassium pump. I can't overemphasize the energy requirements of the sodium potassium pump, because that's where it takes 2/3 of the energy delivered to the brain to drive. I mean, it may be 70%, maybe 60%, people estimate 2/3, to just drive the sodium potassium pumps to maintain that equilibrium. Then action potential, synaptic potentials, and current dipoles. We're interested primarily in synaptic potentials. That's what generates the signals that we're interested in, are the synaptic potentials. In very few instances do we actually record action potentials. In very few instances do we record activity from the inner neurons. It's usually activity from the pyramidal neurons related to the synaptic activity. Very important. And then the activity that we see is volume conducted from the pyramidal cells to the scalp. So it's this conductive milieu of the ionic bath, which we are, that produces the ability to have this volume conduction, because the fields are set up by the ion concentrations along the pyramidal neurons, and that produces the spreads of these current flows, which then show up as voltages on the brain. On the surface of the scalp, we call them EEGs. On the cortex, we call them cortograms. And inside the brain, we call them local field potentials. But they're all set up the same way. So that's this section. So well,
It's one of the most complicated part for